So this is a very happy strawberry plant after picking the strawberries. So we see a tray with growing medium and we see a very big plant on it. So here you see, one of the issues we have with this kind of trials, if you want to look at the nutrient balance, or we want to figure out if there is a balanced nutrient supply, we need to sample the nutrients in the above ground vegetative biomass, and we need to sample the nutrients and the spent horticultural substrate to see actually what is the balance between the two of them. And you can look at it from a crop perspective, and then it's mainly important to see whether the EC or the pH, they are still okay for growing a crop, or if the nutrient supply is balanced for having a good crop. However, when you look from a sustainability point of view, it's mainly important to have really a good nutrient balance. It means that the amount of nutrients which are left in the spent growing media, there should be really low compared to the nutrients that you have used for the strawberries and which remain in the plant parts. Otherwise, you will remove quite a lot of nutrients with the spent growing media, and you lose these nutrients, or they can give problems for environment if they are not properly handled. So nutrients, they are really the more challenging part for this kind of trials, and also for soilless cultivation in general. And well, when we look at nutrient retention and the biomass, we need to assess the nutrient contents on a dry matter basis, for instance, and then multiply it with the dry biomass of the plants. And well, mainly the biomass, it can be quite variable from trial to trial and from greenhouse to greenhouse, depending on a lot of conditions. And then the nutrient retention in the spent growing media, it's also measuring the total nutrient content and the growing medium and multiply it with the dry mass. And also here it can be quite variable depending on how much material is there in the tray and also the composition of this material. So the nutrient balance of this strawberry cultivation, well, we have import of nutrients. There are nutrients which are already present when we start a new cultivation with the growing medium. They can be from fertilizers or from materials. And of course, during the trial or the crop cultivation, you will add nutrients with the fertigation. You have also export of nutrients, so you pick fruits and you harvest the nutrients in these fruits. And there is nutrient export with the drain, so these are like the input and the export of nutrients. And then you have the nutrients which are stored in the trial at the end of the cultivation. So we have the nutrients in the plants, and we have the nutrients in the spent growing media, and of course also in the roots, which are also in the spent growing media. And this balance between above ground and below ground, it's really depending on the cultivation and also on the crop management. So here we have a graph showing the nutrient contents in strawberry leaves. These are the green ones. Then we have also measured it in quite a lot of compost samples. That's the brown ones. And then we have the blue ones. They are the spent horticultural substrates. And the red or the yellow ones, they are the virgin materials that we use in growing media, like peat, coir, wood fiber, or other plant fibers. And here we clearly see that, for instance, for nitrogen, phosphorus, and potassium, we see that the nutrient contents in strawberry leaves, they are very high compared to the other materials. And we also see that the spent growing media, they are, compared to compost, they are lower, but they are sometimes, higher than in the virgin materials. Calcium is like the exception on this rule. Here we see that spent growing media, but mainly compost, they are very rich in calcium, and the calcium content of strawberry plants, it's lower. It's because strawberry plants cannot take up that easily calcium from the growing medium. And what we see here actually is that you have always the two in the box plot. You have two colors. You have the dark green and the light green. It's actually an experiment we did about screening with near infrared spectroscopy, if we could use it as an easy screening method for measuring nutrients, because if you analyze it with chemical methods, it takes a lot of money and time. and it works quite well. So we can actually use these techniques for fast screening of nutrients. And then you see the yellow lines, and these are actually the nutrient contents we had in the trials on reusing growing media. And compared to the box plots, we see that what we had in the spent growing media in our trials, it's quite low compared to what we have observed and the overall samples that we have collected from different growers and different trials. So it means, I think that fertigation, it's really key for assessing, or it's determining actually how much nutrients are left in the spent growing media, so how much nutrients you remove with the spent growing media.